Hi, Dr. Crowder here, and this video is to give an example of how we can test statistical significance for genetic data, looking at examples specifically for linkage between two genes. And as we know, genetics is a quantitative science, and so we need to be able to analyze our data with statistics to determine whether the results we have are statistically significant and whether we can say that our data then supports or does not support our hypotheses. One statistical test we can do to analyze genetic data is a chi-square test, which really is a test looking at what is the goodness of fit of our data. And it's used to determine if the deviations in our data are caused by sampling or experimental error, if they're caused just by random chance, or if the differences in our data are due to a statistically significant real difference, in which case it gives us evidence to support or reject our hypotheses. And normally when you're approaching a chi-square test, you start with your null hypotheses that there is no difference in the data, meaning that any differences you would see are really just by sampling errors, experimental errors, or by chance. And so we want to calculate our chi-squared to generate statistical support for whether we can reject that hypothesis and say, no, the differences we're seeing in our data are statistically significant, they are real differences, and if so, we would then reject the null hypothesis and accept our alternative hypothesis. And I'm showing you here the formula for how you calculate chi-squared, where it's the sum for all events of the observed frequency of a given event minus the frequency you would expect for that given event squared divided by the frequency you would expect for that given event. And so we're going to walk through an example of how we do this, but before we do that, what we would do after calculating our chi-squared is go to our critical values of chi-squared table and look up whether our chi-squared value supports a statistically significant difference in the data, whether the probability of a difference being statistically significant. And for this, we first need to look at what our degrees of freedom are, where degrees of freedom is equal to n minus 1, where n is the number of different categories you are analyzing or comparing in your data, or it's the number of different independent variables or independent events you are comparing for a difference. So n is the number of different independent events you are comparing, and the degrees of freedom is n minus 1. And when you're analyzing your chi-squared value, you would look up then your degrees of freedom. And when analyzing biological samples, there tends to be a consensus that a p-value of 0.05 or smaller supports statistically significant differences. So what I mean by that is if you have, let's say, a degree of freedom of 1, and your chi-squared value is 4, that means your chi-square value is somewhere around here. In which case, your p-value is going to be between 0.05 and 0.025%, meaning that the p-value of 0.05 is 5% probability or 0.025 is 2.5%. So what that tells us then is that in our data, there's between there's a 5 to 2.5% chance that the changes we are seeing are due to chance and not due to real difference. And so what we say is if it is a 5% chance or less that it is just due to chance, due to random sampling, we would say that that is enough support to reject our null hypothesis and accept the alternative that yes, there is a real difference in our data. If our chi-squared value falls somewhere where the p-value is greater than 0.05, so 0.1, 0.5, 0.9, this is saying, for example, right here, this is saying that there's a 50% chance that the difference we're observing is just by random sampling or by random chance. Here it would be by 90% and so on. And so we're saying if the chance that it's due by random sampling or random chance is greater than 5%, that is not statistically significant, in which case we need to accept our null hypothesis. So let's apply this now to an example of linkage between two genes. And so we're looking at two genes, and I'm giving you one as eyeless, where eyeless flies don't have eyes, and miniature wings, where miniature flies that are homozygous recessive for this allele have tiny wings or miniature wings. And I'm telling you that you cross wild-type flies to a double mutant fly for eyeless and miniature, then perform a test cross with the F1 dihybrid you would generate from that cross and get the following F2 progeny. And I'm asking then, based on these progeny numbers, can we determine whether these genes are linked or not? And what is the associated p-value that would then support or not support linkage? So now we're going to use chi-squared to determine, based on this data, whether the eyeless and the miniature wings genes are linked. So our null hypothesis would be that they're unlinked. Okay. And our alternative hypothesis then would be that they're linked. And these are our observed results right here for the 4 different progeny phenotype classes. If our genes are indeed unlinked, based on what we know about the law of independent assortment of unlinked genes, we would expect a 1 to 1 to 1 to 1 ratio of progeny phenotype classes. And since we have a total of 2, in this case, we would expect to see, if these are truly unlinked genes, 50 progeny for each class. So this is our expected. Okay. So let's calculate chi-squared then. To calculate chi-squared, we need to add up the sum of the observed minus expected squared divided by expected for every class. So for the wild-type class, our observed is 53 minus 50 expected if it was segregation, independent assortment of 2 unlinked genes, divided by 50. For the eyeless phenotypic class, it would be 44 minus 50 squared, divided by 50. For the miniature wing phenotype class, 46 minus 50 squared, where we have the number of miniature wings progeny we observed minus what we would expect. And then for the eyeless miniature wing double, our observed is 57, and our expected would be 50. Okay. So if you do the correct math then, these are the numbers that you come up with, resulting in a chi-squared value of 2.2. So that is our chi-squared value for this data. Now before we go to our chi-squared table to look up that number, we need to know our degrees of freedom. In this case, we have looked at 1, 2, 3, 4 different independent events happening. So each independent event here are the 4 different progeny classes. So our degrees of freedom is 4 minus 1, or 3. Because we have 4 different independent events or independent variables that we're considering. Each independent variable is a different phenotype based on the segregation of these 2 genes, which we're determining whether they're linked or not. So let's go and look up our chi-squared value. We calculated that our chi-squared value is 2.2, and our degrees of freedom is 3. So if we look up the degrees of freedom 3, and we look for our chi-squared value of 2.2, it is somewhere in between p equals 0.9 and p5, because here it's a chi-squared value of 0.584 and here is 2.3. So our chi-squared value is closer to p is of 0.5. And what we just went over, that statistically significant data for biological samples is when p is equal to 0.05 or less. So what our data tells us, that based on our chi-squared value of 2.2 with our degrees of freedom, the likelihood that our data, the difference in our data of our observed from our expected progeny, the differences that we observed could possibly do by chance probably about 50% of the time. So 50% of the differences we've observed, or 50% of the time, those differences are probably by chance. And that really is not strong enough to support the alternative hypothesis that there are statistically significant differences we're observing. So in this case, we would accept the null hypothesis, where our null hypothesis were that these two genes are not linked. So let's return to our original question, asking whether eyeless and miniature genes are unlinked. Based on our data, we had a p-value that is greater than 0.05. In fact, our p-value was actually greater than 0.5. And so what that tells us then is that we would accept the null hypothesis that the genes are unlinked, in which case we would reject our alternative hypothesis that the genes are linked. So no, the genes are not linked, and we know that is a statistically supported answer because our p-value is greater than 0.05. Had our p-value been less than 0.05, for example, answer choice A here, that would have supported us to reject the null hypothesis and accept the alternative hypothesis that yes, the genes are linked and it's statistically significant. Our data is statistically significant in supporting that conclusion. So hopefully going through this example gives you a better idea of how you can use chi-squared to support genetic data when analyzing different hypotheses for changes from between what you would observe and expect for your data.